Welcome to Cetacean Sessions.
All right. Hello, everybody. Welcome to Cetacean Sessions. Hi to everyone on the Facebook event as well. We're really excited to have you guys today with us. And yeah, we're very excited to have Tommy present some of his research on bowhead whales in the Canadian Arctic. So yeah, I'm going to get started right now and bring Tommy up. So Tommy is an early career researcher working with the Department of Fisheries and Oceans in Canada. And he just did his master thesis on bowhead whales in the Canadian Arctic. So he's going to get to tell you a lot about that. And he's also going to present some unpublished results with us, which is really exciting. So I'll bring Tommy up. Hi, Tommy.
Hello, how's it going?
Good, how are you?
Good. Thanks for having me.
Yes, of course. So you're not actually in Winnipeg anymore. Where are you at the moment?
I'm based out of Kamloops in beautiful British Columbia, Canada. So yeah, moved here a few months ago for this job with Fisheries and Oceans. Yeah, really happy to be somewhere new once again.
That's amazing. And what do you do right now with Fisheries and Oceans Canada?
So I work on Fraser River Chinook or king salmon stock assessment. So yeah, taking a break from marine mammal and Arctic stuff to do some very applied resource management fisheries. So trying something new. It's been really good so far. So I'm excited to keep going that route.
Very cool. That's wonderful. Yeah. So thank you so much for telling us. I'm quickly going to remind everyone of how everything works before we get started with your presentation. So you guys have access down under the window. For people on Crowdcast, you have access to a bunch of different tabs at the bottom of your screen. So you can click on the Ask a Question during the presentation if you have any questions to ask to Tommy. They could be questions about bowhead whales. They could be questions about his current job. It could be questions about the life of an early career researcher or the life of a federal employee, et cetera, et cetera. And once someone asks the question, everybody else can upvote or downvote this question. And obviously, we'll get started with the questions that have the most upvotes. So when the person who asked the question, when that question gets voted, we'll invite you to come on screen with us to ask Tommy your question. It is totally optional. If you do not wish to come on screen, you can just decline the invitation and we'll ask the question for you. So yeah, for the people in the Facebook Live, you can also ask questions, but you won't be able to come on the Crowdcast Live to ask your question. I posted a link on the Facebook event, so if you want to join us on Crowdcast, you're invited to. And otherwise, we'll keep monitoring the Facebook chat and ask the questions for you. And the last thing, if there's any issue during the streaming with the quality of streaming, or if anything freezes, you're invited to just refresh the page and it should take care of the issue. So yeah, I think that's about it. Naomi, did I forget anything?
I think you covered it all. We'll be monitoring, like you said, the chat as well. So if we do see something's gone awry, we'll try to fix it on our end as well.
Yeah. Yeah. All right. Well, I think Naomi and I are just going to mute ourselves and let Tommy present. And we'll be back soon with you.
And yeah, let me know if anything at some point goes wrong. Just don't forget, don't feel bad to interrupt me, and we'll adjust.
Sounds good.
All right, can you see my slide?
Yep, we can see everything.
Wonderful. Just gonna get a little timer going to make sure I don't go too much over time. Yeah, thank you so much, everyone, for being present today for my talk with the title being Big Appetites, Unique Tastes, a look at bowhead whales feeding in the Canadian Arctic. So yeah, today the focus is going to be bowhead whales, but I'm going to start with a little background about myself and what I currently do. So as I said earlier, I'm currently a stock assessment biologist for Fisheries and Oceans Canada, so working on Chinook or king salmon stock assessment from the Fraser River in British Columbia. So taking a little break from Arctic stuff, from marine mammals. However, it's very closely related still to marine mammals because Fraser River Chinook is actually a big component of the diet of southern resident killer whales in BC. There's like a lot of co-management between like the fisheries and that species of salmon paired with the southern resident killer whales. So still some marine mammal stuff related to it. So yeah, I did my undergrad in biology at Laval University in Quebec City, and during my first semester there, I got a little job in a lab, and my job was to do zooplankton taxonomy. So spend just countless hours looking at those little things in front of you right now, identifying the species, which was great. But then I worked hard during my undergrad, got good grades, and then got some scholarships, which allowed me to go sample those copepods. And this is where I ended up for 2 consecutive summers. So I worked on the Canadian Coast Guard ship, the Amundsen, which is an icebreaker that is based out of Quebec City. So in the winter, it's just de-icing the maritime route in the St. Lawrence. But in the summer, when there's no ice in the St. Lawrence, it's used for scientific expeditions in the Arctic. And you might recognize it for my fellow Canadians because it's on the $50 bills. So after my undergrad, doing those like early, like getting, I guess, immersed in that research in the Arctic, I was hooked and I was like, okay, I want to keep going. And as you can probably tell from my lovely French accent, English is my second language. And I knew that if I wanted to pursue research, my English would have to be pretty strong. So I just thought I'm just going to go do my master's in another province and, get fully immersed in the like scientific research in English right away. And I found that Winnipeg in the Canadian prairies, out of all places, is actually a pretty big hub for Arctic research in like marine mammals, as well as a bunch of other things. So I ended up doing my master's on bowhead whales at the University of Manitoba, and I was supervised by Stephen Ferguson, who is a research scientist at Fisheries and Oceans Canada in Winnipeg, and just a marine mammal legend. He's published over like 100 publications probably on marine mammals in the Arctic. And my work was based out of Igloolik in Nunavut, which is in the Canadian Arctic. So today what I'm presenting is mostly some bowhead research. That was the first chapter of my thesis, and it was on the differences in individual habitat use, foraging dive depth, and diet during the peak feeding season. So this article was recently published about like 2 months ago now. It's in open access, and the link is also in the chat. So if you want to go read it afterwards, you're welcome to. And then you can also contact me after if you have any questions regarding that. So we'll give you a little bit of background. So we can talk about the Arctic without talking about climate change. And under climate change, the Arctic is one of the fastest changing environments. So all those changes are affecting the sea ice dynamics specifically. So we see an earlier melt and a later formation of the sea ice cover, as well as a decrease in the minimum summer extent of the sea ice, which is affecting ecological dynamics at all trophic levels. So specifically for zooplankton, which I studied during my undergrad and was still related to that in my master's research, those changes in the Arctic marine environment can affect zooplankton in many different ways, affecting developmental rates or their phenology, so the timing of certain life history events, which is crucial in the Arctic because we get like very like pulses of productivity. So in the spring, there's like a big pulse of phytoplankton productivity, but then the rest of the year is pretty calm. So timing certain life history events with that spring bloom is quite crucial. So if the sea ice dynamics are changing, then it can also affect the phenology of zooplankton species. The changes in the physical environment can also push species out of their physiological tolerance range, which can kind of rearrange community compositions locally. And we also see distribution shifts. So those like temperate species can extend their distribution further north, whereas those polar species are kind of been pushed and restricted towards the poles. And all those changes in zooplankton, well, obviously they have impacts on higher trophic levels and on planktivorous, carnivorous predators like bowhead whales. So bowheads, they are mysticetes, so baleen whales as opposed to toothed whales. And they are the only whale species of the genus Balena and also the only Arctic endemic baleen whale. They are easily recognizable with the absence of dorsal fin. The white unpigmented patches that are located on their chin and their fluke notch, their large head that takes about like a third of their body, their paddle-like pectoral fins, and they are also the longest living mammal at over 200 years of age. They can reach lengths of like 19 meters and around 100 metric ton, which by weight makes them the second largest whale just after blue whales. So they are super efficient filter feeders, and their filtering apparatus consists of around 600 baleen plates that are all attached on a narrow and arched rostrum, which gives bowhead whales their name. And just to give you an idea of the scale, the size of the whales, this is me inside the jaws of a whale that was harvested a few years back. So when I went to Igloolik this summer, we could go see that. So you can tell it's just huge animals. So there are 4 distinct populations of bowhead whales. They have a circumpolar distribution, so all in Arctic waters. And one specific population that I was interested in for my master's thesis was the Eastern Canada and West Greenland population, which, as its name suggests, located on the Eastern Canadian Arctic and on the west coast of Greenland. So in the summer, their distribution is mostly around Baffin Island in those red zones that are highlighted on the map. And so they're going in different foraging areas around their distribution range. But in the winter, as the sea ice is starting to like retreat south, they are more restricted in the southern part of their range, so mostly in Hudson Strait. So when facing those changes in prey that are driven by climate change, there are like 2 main ways in which bowhead whales could respond. So first of all, there could be evolution, so evolutionary adaptation between generations to kind of try to cope with those changes in prey. However, with a relatively small population size, so there's only 10,000 individuals approximately in that population, a slow growth rate, which results in a late sexual maturity at around 25 years of age. So therefore, like a long generation time, the rate of potential evolution is going to be like greatly exceeded by the rate of changes that we're seeing in the Arctic right now and in zooplankton assemblages. So secondly, they'll have to rely on their ecological plasticity, so their ability within a generation to try to adapt and cope with these changes. They can do this in many ways, and with good habitat connectivity across their distribution range, and also their very high mobility. So they like migrate thousands of kilometers every year. So they're able to like they could be able to track changes in prey assemblages. We also see some variation in habitat use by different demographic groups. So depending if it's like a mother with a calf, or like young immature males or mature females, they're like distributed in different parts of the range. And finally, we also observe some diet variability within the population. So seems to be some good potential like on that ecological plasticity aspect to cope with changes in prey. And as you can imagine, studying diet variation on a huge Arctic whale could be challenging. However, we have some great tools that we can use that allow us to get dietary information with like minimal invasive methods. So basically, we're taking tissue samples, so usually skin and a bit of blubber, and using dietary biomarkers to get some information about the diet. So I'm going to explain some of those that I used in this research. I'll try to keep it as accessible as possible. It's not super intuitive, but we'll try to go through it. So the first approach is by using stable isotope analysis. So I'll try to break it down. So an isotope is basically just different form of an element so that they change. So for a same element, they can have different numbers of neutrons. So they're going to keep the same charge, but they'll have different atomic masses. Stable refers to the fact that they're not radioactive. So for example, carbon-14 is a radioactive isotope that can be used as some of you might know for like dating certain like fossils or whatever. So we don't want that. We don't want the nucleus to be decaying. We want it to be stable. So we're using the stable isotopes. And then it's expressed as a ratio of the heavy form compared to the natural, more abundant form, and using that little symbol, the delta, that you'll see again in this presentation. But basically, the premise behind it is kind of you are what you eat. And the way I like to explain it is your tissues, they are physically built with molecules that you have to acquire from your diet. Like those molecules are the building blocks for your molecules in your body to build your tissues. So if you're measuring the ratios of stable isotopes in a predator, well, they're going to be the result of the stable isotopes that were found in the prey that they ingested, but modified by some fractionation that we call. So basically just different biochemical processes that change depending on the fact that those isotopes have different atomic masses. I'm sorry if that was a lot. This is actually going to help to illustrate that. So usually we use nitrogen and carbon, and they're usually showed in a isotopic biplot like this one. So on the y-axis, we have nitrogen, which helps to distinguish different trophic levels. So those lower values are like your phytoplankton, zooplankton, and then higher values will be your top predators, so like killer whales. And then for carbon, it's different carbon sources will depend, like they'll go left or right on the scale. So if you measure your stable isotopes in two distinct individuals and they have like different stable isotope values and are like separated in that biplot, it means that they likely fed on different prey assemblages. So the closer they are, the more similar their diet is, and the like further apart they are, the more different it is. And another approach is by using fatty acids. So fatty acids, they are the simplest lipid forms. So those building blocks to build fat. And they're like a very simple molecule. As you can see, it's just like pretty linear. So it's a long carbon chain with a methyl end and then a carboxyl acid group at the end. And they can vary in two main ways. So the length of the chain and also the presence, position, and number of double carbon bonds in between them. You don't have to remember that. The only thing you have to remember is that is very useful for us is that those dietary fatty acids, they are thought to be incorporated without modification in carnivorous marine mammals. So what that means is that if you have a specific fatty acid that is found in a specific prey, well, it will be found intact, so no modification to it, in a predator that consumed in the fat like of the predator that consumes such prey. Meaning that if you compare the fatty acid profiles, so like the different assemblages of fatty acids in the fat of two different individuals, and if you see differences between those, well, it means that they likely fed on different prey. So super useful tool that you can all get from just like a tiny skin and blubber sample. So previous studies have used those techniques to look at inter-individual variation in the diet of the same bowhead whales, like the same population that I looked at. And as you can see from those graphs, there's some clear groupings of individuals with similar like diets or dietary biomarkers. However, none of the groups were dominated by individuals of a specific sex, age, year, or region. So basically the mechanism that's driving this grouping is still unknown. So it's like, what is that mechanism driving those differences? That was the objective of this study, to investigate potential drivers of diet variation among bowhead whales from this population. And my hypothesis was that foraging behavior is what is driving that dietary variation. So not the sex of the individuals, not their age, not where they are, but more their behavior. So my hypothesis was that differences in horizontal movements, so the habitat that they use and where they go, as well as vertical movements, so how they dive to acquire their food during the peak feeding season, will be what is driving that difference in dietary biomarkers between the individuals. So our study design, I used archive data. So I started my master's in the pandemic, so couldn't go collect data, which was quite unfortunate, but kind of taught me, I guess, the value of going back in your archive data and trying to do something cool with it, which I think this is a great example of. So I had access to 8 whales that were tagged and biopsied in Igloolik in 2012 and 2013, and then another 8 whales that were tagged and biopsied in Pengnirtung in 2012 and 2016. And I've done this presentation a few times, and people sometimes get confused. Even if they're like in different part of the range in the summer, they are part of the same population. Just want to make that clear, but they just use different habitats during the summer. So the whales, they were tagged and equipped with a satellite telemetry and time depth recorder tags. So this allows us to track their horizontal movements between the regions, so where they're going and when they are at those locations, as well as vertical movements in the water column, so what they're doing when they're underwater searching for prey. And we restricted our analysis to the peak feeding season, so from July to November. We were also restricted by how long the tags last, so that was kind of the sweet spot to keep looking at it. And then the whales were also biopsied when they were tagged, so we could get readings on stable isotopes and fatty acids, which I explained to you earlier on. And then the analysis was a simple general linear model, which I'm not going to get into. Don't have much time to go through it right now, but if you're interested in it, you can read the paper. But basically trying to like see which predictors are explaining that variation in the diet that we're seeing. So I'm just going to go through the main results. So the whales that were tagged and biopsied in Igloolik, so the white square, they used the Canadian Arctic archipelago habitat, which is highlighted in blue here. And after being tagged, they performed very long distance seasonal movements. So the different arrows are just the general movements, and the different colors are by month. So you have July, August, September, October, and November. I'm just going to walk you through it quickly. So after being tagged here, they headed west through Fury and Hecla Strait, and then spent most of July, August, September, and October in Prince Regent Inlet and Gulf of Boothia, which is a big foraging ground for this population. But it's at the end, like towards November, that something quite interesting happened. So 4 of those whales went north, like around Baffin Island, and circumnavigated it, and then headed south towards Hudson Strait at the end of November, whereas some of them just went back through Fury and Eccles Strait, through Fox Basin to go towards Hudson Strait as the sea ice is starting to go south. So that was for the whales that used this habitat. Then we identified another key habitat, which was the West Baffin Bay. And the whales that used this habitat were mostly the whales that were tagged around there, so tagged in Penguituk. However, their horizontal movement patterns were quite different. They didn't move a lot. They stayed in that little inlet there that's called Cumberland Sound for most of their periods. So from August to September, October, stayed pretty stationary in that little inlet and only headed south towards Hudson Strait once again as the winter was approaching. So those differences in habitat use and horizontal movements were also reflected in the dietary biomarkers of the whales. So as you can see here, that's our 8 whales that used the Canadian Arctic Archipelago habitat at higher isotopic values and are shown in the upright corner of the graph, whereas the whales that use the West Baffin Bay habitat had lower isotopic values and were shown more in the bottom left. So we see a pretty clear separation between the two groups using distinct habitats, which was great news for us. And the same was also found in a specific fatty acid ratio that we found that was higher for all the whales that use the Canadian Arctic Archipelago habitat compared to the West Baffin Bay habitat. So kind of showing that there might be some differences in diet between those whales that are using distinct habitats. And by using some previous data that was collected a few years before that those whales were tagged and biopsied, we could see that the zooplankton species that are part of the bowhead diet in that area, some of them have those higher isotopic ratios. So the blue whales that we see here from the Canadian Arctic Archipelago likely fed a bit more on those higher trophic level zooplankton species, whereas the yellow whales likely fed more on those lower isotopically enriched prey species that were the smaller copepods, whereas those bigger ones are the big euphosids, krill. Parachaeta glacialis is a big predator of those smaller copepods, and same thing for Chaetagnath, so that's why they're higher up on the nitrogen scale. And then the clear differences that we saw were also reflected in the vertical movements of the whales. So looking at their foraging dive depth, so each box here is an individual, and all the dives that they perform from the period of July to November are all together. So we see that the whales that use the Canadian Arctic Archipelago in general add shallower dives than the whales using the West Baffin Bay habitat, which right here, that's something we discussed in the paper. Bathymetry obviously played a role here, because how deep you can dive depends on how deep the water is. So there's some of that here, but there's also some cool behavioral differences between the whales using the two habitats that are more likely, not more likely, or just more easily seen when we pool all the individuals together that use the different habitats and look at the foraging dive depth by month. So we see two quite distinct patterns. So for the whales using the Canadian Arctic Archipelago in blue, we see this increase in foraging dive depth as the months go by, whereas for the West Baffin Bay whales, it's more consistent in their foraging dive depth. So the West Baffin Bay whales, if you remember well, they stayed mostly stationary in that little inlet, so Cumberland Sound. So they likely more reliably found a patch of good prey at 250-ish meters, and were just targeting it for all the months, and it was reliable, so they didn't have to, to adjust too much. Whereas the whales in the Canadian Arctic Archipelago, that super cool increase in foraging dive depth is actually a behavioral response to vertical movement of their prey. So copepods, and more specifically Calanus copepod, they have this super cool life cycle that in the winter they enter a diapause. So it's a form of dormancy that you just go at depth near the the seafloor, and then they just don't move a lot. And then they're gonna ascend and go up the water column in the spring, trying to match their ascension to with the spring bloom, as I talked earlier, like the phenology aspect, that's where it's important, and trying to produce their eggs at the right moment so their like little copepods can feed on the spring bloom and then develop and have like those bigger, like basically acquire a lot of fat to build that inner energy storage, and then that they need to enter their diapause for the winter. And that movement that we see here, like summer, fall, and winter of the copepods kind of like descending at depth, is what we're seeing, is the behavioral response we're seeing in the bowheads, that they're like following this like vertical migration of their preferred prey. So like super cool. So overall, yeah, saw quite clear differences between whales using distinct habitats. They're still part of the same population once again, but they're just using very distinct habitats. And we saw like quite clear differences in their dietary biomarkers that were likely associated with differences in diet. And also like clear differences in foraging behavior with horizontal movements, either like doing very long migrations or staying mostly stationary, having like shallower dives on one hand for the Canadian Arctic archipelago whales compared to like deeper foraging dives, and then this cool monthly increase in foraging dive depth or like monthly consistency. So like strong association between foraging behavior and so habitat use and foraging dive depth, and also the dietary biomarkers. So our hypothesis was supported. So foraging behavior was explaining some of the dietary biomarker variation that we were seeing. However, we didn't, like one of the limitations, I guess, is that we didn't sample prey. Well, I wasn't there when I was in high school back then when they did that, but they didn't sample prey. So we can't fully associate those differences in dietary biomarkers with specific preys. But it's still like a good sign that they're doing something different and that there's this cool like differences in foraging behavior. Okay, doing good on time. So that was the first study that I had, so the paper that was published, and you can go read about it, and I'll be happy to take any questions on that. But the second chapter of my thesis was looking at another aspect of it, and this is unpublished. We're still working on it. I'm going to be submitting it probably within the next month to a journal. It was to look at individual specialization as a potential driver of diet differences. So basically the individuals themselves being specialized in maybe like in like acquiring certain types of prey. And to look at that, we used a super cool approach that we use baleen plates from bowhead whales. So like part of their filtering apparatus, as I showed you earlier. So Inuit people in Nunavut, as well as in Greenland, they have the right to hunt bowhead whales for subsistence, and they're also doing it sustainably. And part of that agreement is also to provide samples for science. So some of the whales that were harvested between 1998 and 2011, so there were 19 of them that were kind of like spread across the range of the population, as you can see on the map. And we tried to collect the longest baleen plate from each harvested whale. And baleen plate, they're super cool because they are made from keratin. So it's the same proteins that your hair is made of, which means that they are metabolically inert after their synthesis. So once like part of the baleen plate is formed, it will not like interact with the rest of the body, and then it won't change. So if we're using stable isotopes once again, like this whole baleen plate is basically a timeline of changes in stable isotopes over time, which allows us to quantify how those stable isotopes are changing within that individual over time, and also compare between individuals how they're changing over time. So this is what the isotopic data from a typical baleen plate looks like for both carbon and nitrogen. We see quite clear cycles that are actually annual cycles. They're not always super clear, and that was kind of a struggle when you go into it, like attribute a year to every cycle. But mostly annual. And for carbon stable isotopes, it's actually tracking those like big migrations that they're doing throughout their range. It's been like incorporated in the baleen plate as it's growing. So we see like those movements. And for nitrogen, it's tracking variable food intake. So all the peak values are the peak summer values when they're like consuming a lot of prey, and the low values or the lower when they're not consuming a lot of prey in the winter. So when you're taking all of those like little data points from carbon and nitrogen and putting it back in this biplot that I showed you earlier, that's kind of how you're trying to see if there's a specialization within the population. So this is an example, this is a hypothetical example of that variation in stable isotopes between the individuals is greater than the variation within individuals. So each individual in that case would be more like specialized on a different like type of prey over time. So you see that they're what we call the isotopic niche. So like the little ellipse around it, around the individuals are like quite separated. So that would be an example of specialization. But on the other end, if the ellipses are like super wide, so there's more like variation within each individual, and the ellipses are like all overlapping as they're doing right now, it would be an example of more like generalization in the population and like less specialization, basically. So we did run some fancy models to look at that and quantify that variation within and among individuals. And the results showed that most of the variation for both carbon isotopes and nitrogen isotopes is associated with variation within individuals more than among individuals. So basically, we're not seeing any signs of like super strong individual specialization in the population. And if we look at the actual data, this looks kind of a mess because we have a lot of data points. But once again, all the ellipses, each ellipse is an individual with its isotopic niche. And we see a lot of overlap. So no sign of like clear individual specialization, basically, on that front. So that variation within is greater than among individuals. So this is still work in progress. We're working hard on the discussion right now, trying to like see what that means as well in the like broader ecological context and like with climate change and everything and their resilience. But it's a super cool approach. So keep an eye out for that work in the upcoming months. So the overall conclusions from my thesis, once again, in that context of like changing preys, composition and abundance with climate change, is that there seems to be good individual level plasticity of bowhead whales from that population. So either if it's like behavioral or differences in their habitat use or differences in diet, there's like some good variation that we see among individuals that are using distinct habitats, for example, and then also within individuals, as we saw from the baleen plate records. So all that variation within and among all the different individuals, when you like sum it up together at the population level, well, what you get is resilience, basically. So some individuals might be, more negatively affected by certain changes in prey, because if it's like their preferred prey or something that is being more affected, then they might be more, negatively affected. But it's, once again, kind of like that portfolio effect of if you have a lot of variation within your population, between your different individuals, you're getting resilience to cope with those changes in prey that are driven by climate change. All right, that's pretty good on time. Thank you so much for listening. This is our crew this summer in Igloo Lake on a sketchy piece of ice, tagging and taking biopsies from bowheads. And different. So this project was funded by a lot of different agencies that are all listed here. And thank you so much to my collaborators or like local Inuit research assistants on the field, as well as anybody who helped me go through that process. And I'll be happy to take any questions at this point. So I'll go back to the platform. And I think I have to stop sharing.
Hold on, I'm just bringing Naomi back and unmuting her. Okay. Wow, Tommy, I have to say, I have never seen someone present stable isotopes and fatty acids and the implications of them with like such ease. Yeah. And I think that anybody who has ever like approached stable isotopes would be very confused, but you have made it so easy and so clear. Thank you so much, because that was just incredible.
Thank you.
Yes,
Yeah,
thank you.
I appreciate that. Yeah, it's tricky, and try to keep it short and sweet and make it accessible. But yeah,
No, I think you did
Definitely.
an amazing job at it.
yeah, thank you.
Definitely great job. And your graphics, like your pictures and everything, that your little tiny bowhead whales, beautiful. It explained things so well too visually. Perfect.
Yeah. Yeah, it's mostly because my parents attended my defense, and I wanted to make sure they understand. So I was
I
like,
love that.
we'll make it really nice for them, and then I just kept it like that.
Nice. No, that's good.
Yeah, that's for sure. That's how I started with science communication too. That was for my mom. So it's always nice, but I think the graphs made it super clear. I really liked how you did the biplot with the generalist versus specialist niches. I thought that was really, really cool. So I see that we have a couple of comments, and Naomi and I are going to ask you the comments directly, because the person does not want to appear on screen, which is totally fine. But Naomi, do you want to ask the first question?
Sure. On the different foraging behavior between the CAA and WBB regions, was there overlap of individual in these regions, or were there two different groups of individuals utilizing each habitat?
Good question. I would say it was mostly, yeah, it's tricky because it was also different years. So it would be hard to say, but it seems like it's quite, they don't overlap too much. There was like those whales from the like Canadian Arctic archipelago that went like around Baffin Island, and then went on that side of Cumberland Sound that could kind of overlap. But mostly, yeah, they're like quite separated, and it's like quite long distances, like between like Igloolik and Pang, you're like 1,000 kilometers away. So it's very in that period of like intense feeding in the summer and fall. It's mostly not overlapping at all, but then they all kind of get together in the winter in Hudson Strait, and that's where they mate as well. So that's why there's like still like this cool, probably like genetic like mixing that's happening in the winter because they all mate. But then like why they're using different habitats, like there's like the different life stages that are using different habitats, but it's still like quite a good question of why are they not going the same places. Yeah, might be actually the cultural learning of over time, like they learned to like use those habitats, and then it was transmitted differently between different lineages. Yeah, something to explore.
Cool, thank you. And that was by Taz, by the way. I forgot to mention who asked the question.
Yeah, thanks for the question. That's great.
All right. Well, we have a second question from Taz, and I'm going to ask it.
Yes.
There were several lovely drone images in your presentation.
Yeah.
What role did drones have in your research?
For me personally, nothing at all. Yeah, but we are definitely using them more also to track, to see. So this summer when we went tagging and biopsying, we were also trying to fly the drone as much as we can to get like some cool behaviors, and then trying to link it with also like hydroacoustic recordings to see like from the air what it looks like, and then like record the sounds at the same time. And also looking at when we're tagging them, like seeing where the tag went, kind of like looking back at it. Then like cool morphometrics. Obviously, you can like measure the whales way easier than just on the fly when you're going super fast in a boat trying to tag it. So there's definitely super cool ways to use them, but personally, it was not. I did not use any drone data for my research.
Cool, thank you.
Thank you so much.
Okay, it looks
all right.
like Gary has a question. How complex was the tagging process? Are the whales wary of vessels considering the history of whaling?
Yeah, good question. It is complex because you have to, it's very tricky because you have to find them, and then there's like ice around, and they're like great at hiding. Like that's the strength of bowheads, is that they love hiding in sea ice, and they can like break the ice with their head to go like breathe. So whenever we were approaching them too quickly, they would just go inside like denser patches of ice, and then we can't really get to them. So it was like trying to get them in the open water, or we were staying at the ice edge and then like shutting our engines off and then waiting for them to come out, and then like trying to go as fast as possible to get them. And also you try to go fast because you don't want to be like chasing them for too long and exhaust them. So it's definitely a very tricky process. That's why also you get like such small sample sizes. Like we were lucky if we would tag more than 2 whales a day, like being 13 hours on the boat, you know? So it's quite something.
Dang, well,
That's
thank you.
really impressive.
Yeah.
All right. Well, next question. We have a Facebook question from Kaim,
Yes.
who said, "Amazing presentation," and I second that. Can you provide further information on the factors that may influence the variations in the individual bowhead whale habitat utilization, foraging dive depth, and diet during the peak feeding season?
That's a loaded question. Yeah.
Sorry about that. Yeah.
That's like a defense question. Okay, interesting. There's definitely a lot, like, and some of them are like anthropogenic sources, and like there's like more like maritime traffic going to the Arctic that is passing through like key like habitats where bowheads are, so it might drive their like utilization of different habitats that are maybe suboptimal for foraging, but they're not being influenced by the ship noise as much. And then there's like way more killer whales. I know like all of you love killer whales, but killer whales, like they get their name from something. It's because they're good at killing, and they're like the main predator of bowhead whales, and they're definitely more and more like seen in the Arctic because there's less sea ice. So it's also driving, like there's some really cool papers about how it's driving the selection of sea ice, like denser sea ice when killer whales are around to like protect themselves. And then might also like impact like their foraging dive depth, where they're gonna go. So there's definitely a lot to cover. Whoever asked that question, if you want to reach out, we can chat about it at length at some point. But that's a really good question.
Yeah, I'm sure Ken will reach out.
Yeah.
Thank you. Then I see we have a question from Val.
Yes.
And I will try to invite Val to the screen so she can ask her question. And it looks like it's not. Okay. Maybe Val, you could tell us in the chat if you don't want to appear, if it's just not working for you. But in the meantime,
yeah,
I'm going to invite Jared on screen. Let's see if it works for him. And if not, I'll just ask his question.
Oh,
Jared, can you tell us in the chat if you get an invitation or not? All right. Well, I'll ask you. Oh, yep, accepted. Okay. Well, it doesn't say that you're coming and you're on your way. So we'll just wait for you another couple of seconds, and if we don't see you.
And Val commented on her question that she doesn't have a webcam on her laptop, and the microphone's not working. So.
All right, that sounds good. And I see Jared down there, but I don't see a camera for him, which I cannot toggle it. So how about, Naomi, do you want to ask Val's question?
Of course, yeah. Is there any impact of sex or age class, adult, juvenile, on your results?
No, actually, and that's like very surprising because so that's also part of the models that we ran, and it's discussed in the paper. But yeah, there's like no effect of sex or age class on the results, which is really weird because there's like some pretty significant like spatial segregation between like demographic groups. So like once again, like mature females are like more found on the west coast of Greenland, and then immature males are like more found in Cumberland Sound, and then females with calves are more found in like Fox Basin because it's a bit more sheltered and a bit more shallow. So you would like probably predict that there would be differences and like an effect of sex and age class, but there isn't, and there hasn't been in also like the previous studies. So it's, yeah, that one is kind of a mystery, honestly.
Interesting. Okay, thank you.
Thank you.
Somebody's on screen. Great.
Hey, you guys. That was
Hello.
a great presentation. Thanks for that. I was just listening to it. We were listening to it here, and I was kind of curious. What do you know if what the mean tag transmission duration was for some of the data that you used?
Yeah, mean transmission, I don't know, but it varies so much. It's like from 2 weeks to like 2 years.
Amazing.
And we have I think 3 whales that had like 2 years of data, which kind of in the paper, if you read about it, we actually use that to kind of validate an assumption that like foraging behavior and habitat use is consistent over time. Mm-hmm. So we use those like 2 years of data to be like, okay, in October of that year, they were around there. And then the October of the next year, were they using the same habitat? And they were. So there's like those 3 whales really helped me to like kind of clarify that assumption. But it varies so much. And we went once again this summer to tag, like we tagged, I think like 14 whales, and we have 9 that are still transmitting. And that's really
and so these are with splash tags, Mark 10s?
No, I think they're splash tags.
Right. I noticed in your paper there were the ones you used for that study, at least, were splash tags. I find it so surprising because I've used these tags in a few places, and they normally last a couple weeks. Maybe you might get a couple months out of them, but why do you think they transmit so long on bowheads in some cases?
Yeah, good question. Okay. I don't know. I know that like this summer we tried to like have them set up differently. Like some of them were set up to transmit like a lot, but for a short period of time. And some of them was like one location a day, but then trying to like last the battery for so long. It's a really good question that I don't have an answer to.
Okay. And are you tagging them right on the top of the back, or?
Yeah, trying to tag them like where the dorsal fin would be, basically.
If they have one, right?
Yeah, exactly.
Okay. Yeah, very interesting. I wonder if it has something to do with cold water. I know that tag transmission durations from Antarctica seem to be a bit longer than other places as well. So.
Yeah, but also it's quite surprising because they're rubbing on ice a lot. So it would also like the opposite would be like more likely of like if it gets off or they manage to like rub it off when they're like going through like dense ice. So yeah, I don't know why some of them just somehow last super long, and it's big data for us. It's amazing.
Yeah, fantastic. Well, thank you.
Thank you.
Thanks so much for the question. That was a super cool question, and I was wondering if it was like maybe because they're not as like going crazy like the humpbacks and like breaching all the time, but if they rub on ice, then
but they do. Yeah, we had a few ones like put on a show this summer, like just like flapping their pectoral. They're like not their pectoral, pectoral they were doing. They're like their tail as well, like tail slapping, then like breaching. So. I don't know.
That's super cool.
Yeah.
All right, then we have a question from Ludovic. I think.
Ludo.
Yeah, I think he's trying to get in. I think. Yeah, there he is.
Hey, how's it going?
Good. How are you guys? Hey, Tommy. Hey, Nice. Hey, Naomi.
Hey, it's good to see you.
So I was wondering about the fact that with decreasing sea ice in the Arctic.
Yep.
So I heard in this. Tell me if I'm completely wrong, but I heard that this potentially could open up new areas that were previously not accessible to feeding. And so I'm sure it's a very complicated answer and a loaded question, but do you have any thoughts about that and how that may affect, distribution of the whales in the Arctic and that?
Yeah, well, totally. Like right now they're kind of restricted to the top of Baffin Island, but there's still a lot of ocean to be seen above that. And yeah, with like, if there's less sea ice cover for like long extended periods of time, like we know that there's like still plenty of food up there. So yeah, it would open up some of those areas for that. And they're also targeting like those like bigger Arctic copepods that are like more like energetically dense. And right now, like at the south, like the southern part of their range, it's mostly like temperate species that are starting to be like more abundant. So if they want to keep like the same energetic value in their diet, like they might have to start feeding more up there. And then slowly distribution is going to shift. And they're like very capable of doing that. Like every year they like, migrate thousands of kilometers. So it's not a little like 500 kilometers, like a bit more north that would change something. So I feel like they would be able to track that for sure.
Yeah, and that raises all kinds of interesting questions too, because as the ice goes away too, there's going to be more ship traffic, right? So
but then they're going to be like definitely more exposed to that. And like that top part of like the Northwest Passage just above like Baffin Island is definitely going to be like a very important shipping lane probably. So how like the Canadian government is going to deal with that is it's a big question.
And it gets more complicated too, because there's Canada, but then there's also the US and then Russia as well. So I think something's going to be interesting how that.
Because the Northwest Passage is actually like from a shipping perspective, it's like more efficient than going to like the Panama Canal for a lot of the shipping that's going on worldwide. So it's definitely a lot of economics and politics involved in that. So it's going to be interesting how those countries deal with this.
Cool. All right, well, really.
But you did mention. Sorry, you did mention resilience in the second part of your presentation, which I think would be very interesting for future research to focus on, considering what we're about to face. It would be very nice to have more studies done on this and confirm resilience or non-resilience for future.
And like regarding this, like this summer, one of my colleagues that was on the field with us was slowly working towards basically a like playback experiment of like ship noise in the Arctic for bowheads. So like trying to like see the impact that like this increased shipping would have on their behaviors and their reactions to it. So that's like something that's in the works right now, and definitely kind of like seeing in the future of how this increase in shipping is going to impact bowheads for sure.
Nice. All right. Well, thank you so much. Thanks, Ludo.
Yes. Good luck, Tommy.
See you around. All right. Then it looks like we have a question about feces.
Yes.
And I'll ask Caroline if she wants to come on screen. We'll see. But yeah, definitely like while we're waiting for her to get on screen, it's definitely huge implications for the future. And we obviously are going to want to know if Arctic marine mammals are capable of going through all the future changes. So it's really cool.
Because Arctic marine mammals evolved in this like very quiet environment with like the sea ice like acting as like a sound barrier as well. So like for the communication and everything, they're relying on a very quiet environment, which is, it's definitely not going to be anymore. So, and at once again, like the evolutionary potential, like, can they be that quick to change how they're like, probably not. So it's how are they going to be able to be plastic about it and like using some, their plasticity to deal around it, or are they just actually going to be really badly affected by it?
All right. Well, it looks like Caroline is not coming on screen, so I'm going to ask the question for her if that's okay. I'm sorry, Caroline. Could collecting feces be done and useful in this type of research?
Useful, yes. Done, we tried, and and we just didn't see any. Like this summer, like we were close to the whales, and there was just no like feces to be collected. We were ready for it because you could do like some really cool like DNA stuff with their feces to like have an idea of what they're eating. It's like even less invasive than getting like a skin sample. But there was just no poop in the water. Like we tried our best, but we just couldn't see it. And like I worked a summer in Newfoundland and I was around humpbacks and we would see them like poop all the time. And like that would have been super easy to do, but I don't know, somehow for bowheads in that region, we just couldn't find any. Yeah.
Maybe they're shy.
That's what Ludo said. Maybe they're shy.
Yeah.
Well, thank you for answering that. And we had a question from the Facebook chat from Rosa, who is curious to know if you have seen any information about potential microplastics being found in the plankton and how this may affect your bowheads.
Yeah, good question. I don't know a lot about this subject. I can't really say. I think the trends that we're seeing in other animals and other aquatic animals will probably be the same in bowheads. And yeah, I don't have that great of an answer for it. I'm sorry, but it would definitely be detrimental. And there are some evidence even in the Arctic, because there's like big, like water circulation patterns in the world, and some of them converge in the Arctic. So even if there's like not a lot of pollution happening like directly in the Arctic, like the pollution in like those like lower latitudes is being kind of pushed towards there. So there's definitely a lot of microplastics even in Arctic Ocean. And yeah, it would be, I don't know, like the implications of it physiologically or anything, but I would say it's probably not positive. Yeah.
Thanks. Naomi, do you have a question for Tommy?
I do not. I was actually answered earlier on. I did write something down during your presentation. However, it was answered. So how about you?
Okay. Yes, I do. I have a ton of questions that I will ask you personally at some point, because I don't want to bore everyone, because they will be extremely detailed. But I was wondering, so you looked at bulk stable isotopes, and you saw these geographical differences between the two subpopulations, at least for this for the summer. I was wondering if you noticed, if you know, like from the literature, there's any baseline variation in those isotope ratios, and if that could explain.
Yeah, there is between those two regions, like the same species. Like let's say like Calanus glacialis, which is a typical Arctic copepod, like that was found in in Baffin Bay or in the archipelago, had like quite some variation. So there's definitely some baseline differences, which like for all the like stable isotope aspect of the paper, that's why we can conclude much, because it's like, well, it's probably all baseline variation. But the fact that it was coupled with like foraging behavior differences and also like more importantly the fatty acids, then we're like, okay, there's probably a dietary component to it. But then we would need to either do like compound specific stable isotopes or get like some mixing models involved and get some really good prey sampling, which could be super doable. Like this summer when we were there like tagging the whales, you have a lot of downtime, and just it doesn't even have to be like quantitative for abundance. You just need like a sample of potential prey to be able to at least compare it at like a reasonable like spatial and temporal scale. So sample prey if you're doing diet stuff. That's my tip of the day.
I agree. Yeah, I definitely agree. Sampling prey is important. Yeah. And I hope that for the future, if you end up or someone else ends up working on the next parts of that project, that they get some cool prey to do some cool stuff with it. So there was a follow-up question about the poop. We're going back to the feces, and it was a suggestion, I think, more than a question, to use a dog to smell the feces, because that's what they used at Wild Orca.
Interesting. I didn't know that.
No.
Okay. We did not use that, and I don't think we thought about that, and I didn't know it was a thing. But I could send that along to my colleagues. Maybe there's a bunch of dogs up there, and we had one.
Maybe we could train them.
Train it to smell, bowhead poop. Yeah.
All right. Well, that's it for the scientific questions. I may ask you one last question before we end. That's about a potential tip for early career researchers, because we have been focusing on early career researchers for this entire season, which is one of the missions of whale scientists. And we'd like to know if you have a specific tip for the early career researchers in the audience.
Yeah, I guess what helped me is, there's so many, but definitely just don't be picky at first. Like when you're starting, just like take whatever comes to you, and then build like a network. And people, like, most things in science is like somebody knows someone, and then you meet somebody at a conference, and then you end up working with them or collaborating with them. So there's a lot of like networking involved. And so like if you ever go to a conference, like take those opportunities to talk to everybody. It doesn't have to be like just about science. Like you can also just be friendly. Like you make friends. Like this summer, that's how Anais and I met. Like we met this summer at a conference on like marine mammals, and then stayed in touch. And then she invited me today, and like today I'm presenting. Other people are like seeing my name, and that's how it happens. So you have to like put yourself out there. I would say that's my best trick, best tip. And then being flexible, like being ready to accept opportunities when they come to you. So like this job that I accepted, like I wasn't even done my master's. It got like offered to me, and I moved like halfway across the country, like on a 2 months notice for it. So I know it's not possible for everybody, but if you can, like, take those opportunities. They're going to lead you somewhere, and it's the time to do it. Like when you're early in your career, and if you're like you don't have like any like too strong attachment to any places. I would say that's like being flexible and being ready to do those sacrifices, because it is a sacrifice. But if you're really into it and that's really what you want to work for, I would say just go for it. Yeah.
Great advice.
All right, well, thank you so much. And thank you for being here. That was honestly like such a good presentation, that I'm a huge feeding ecology fan, and you made the subject very accessible, which I think is hard to do. So thank you so much for participating, and for all of you in the audience, thank you for being here. And don't forget to sign up for the next episode of Cetacean Sessions. It's you should see a little button under the video. Our next episode is going to be on May 8th with two very special people, Dr. Boyd and Dr. Peters, who are both very good at necropsies and cetacean welfare, and how to use necropsies to understand the lives of marine mammals. So really, really cool session coming up. I hope we can see you there, Tommy. And thank you all so much for coming. And we'll be available in the future, like if you have any questions for Tommy, or if you want to contact Tommy directly.
Yeah, actually, I'll put my email in the chat right now.
Thank you.
You can reach out to me. It's also on the paper.
Caroline has also said something in the chat as well, to look up Sam Wasser at University of Washington and conservation canines.
Cool. Thank you so much for that.
Yeah, thank you, Caroline.
Thank you, and I'm sorry we couldn't bring you on the screen. I'm not sure exactly what happened, but thank you for your contribution. And yeah, that's it for us. I'm going to end the broadcast now, but please get in touch if you have additional questions, and don't forget to sign up for the next episode. We're very, very excited about it. And yeah, thanks again, Tommy, and see you all guys very soon.
Ciao.